Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Hello everyone. In previous video we discussed about the introduction, about the measurement of cell characteristics, and three major cell characteristics we'll be discussing, that is cell number and viability, and we are having cell motility and migration, and finally we will be discussing cell functions. So these are the three things we'll be discussing. Actually 5 things we are covering in the 3 topics. Okay, so let's get started. So cell number and viability. Cell number and viability, as the name itself it is there, it's about the cell number of the cells of this particular different cells, and we are having the viability in the sense, cell viability in the sense, so that we already know that the ability of a cell to work, function in different conditions, or after few or over a period of time. So both the properties plays a very important role before you use these cells to seed into the scaffold or a extracellular matrix in order to grow a tissue. So you need to check the cell number and viability. So measuring the number and the viability of the cells are very important parameters before and after the incorporation of the cells in the scaffold. So before implanting or before seeding in the scaffold or before seeding in the extracellular matrix, you take the count of the number of cells, and after seeding, and when the tissue cells gets organized, they start differentiating, differentiating and proliferating and finally becoming a tissue, and then you check the number of the cells. Okay, that need to be checked. So there has to be significant difference, definitely. So or else the whatever number of cells you are giving the same number of the cells are there, that means the cell doesn't undergo for cell differentiation. So if that happens, that means that the cells are resistant to differentiate. So that will not give you a tissue, right? So for that we can see cell number, and we can understand that the cell can have ability to differentiate or not, or can have ability to do the particular function or not. All those things can be done. Now the optimal number of the cells should be seeded for achieving the proper functioning of the scaffolds for tissue engineering, definitely, because we cannot give a very less number of the cells. We should not give very huge number of the cells. There has to be optimal number of cells. That means the number of the cells should be optimum. That means we should know for kidney cells how many cells required, for particular liver cells how many cells required, and we have studied that in the previous topics in the cell number and the growth rate or something like that. So the similar fashion, we have to understand what tissue or what organ we are dealing with, and we have to see what kind of numbers, how many numbers of tissue required, what is the morphology it's required. So here it is about the cell number. So optimal number should be there. What do you mean by optimum? Optimum is something where you are getting the best performance or best yield or best activity. So here optimum number of the cells means the optimum number of the cells, so much number of the cells, imagine n number of the cells is giving you the best function of this particular tissue, then we can call it as an optimal number of cells, right? So thus measuring the number and the viability of the cell is very significant. So similarly we are having similarly to assess the biocompatibility of the material. So if you want to check the biocompatibility of the material and to evaluate their ability to promote the cell division and also cell count and viability at different time after administration is also very important. So if you want to check, now I have these cells, now I want to check its biocompatibility. That means whether the cells are harming any other cells or not, or whether the scaffold, which is a biomaterial, it can be the biomaterial, whether it is having any effect on the cells or adjacent organs. That has to be checked, right? So similarly similar fashion how we discussed about the cell number, similarly the biocompatibility, that's one of the property of a particular material, that is usually the scaffold or extracellular matrix, the synthetic one. Okay, that has to be evaluated to evaluate their ability to promote cell division, whether these scaffolds can provide cell divisions or not. That means whether they will support for cell division or not. So cell count and cell viability, even for that cell count and different viability, that means the how many number of the cells are there. Imagine this biocompatibility, why we are talking about this material is because cell where it will grow on the scaffold. Now what is scaffold? Biomaterial. So it has to be biocompatibility, it has to be biocompatible. And here, how not only the biocompatible, not only it's giving the structural and mechanical support, but also it is giving the cell to adhere, that means attach to it, and then it should be in such a way that the cell should do its function. What is the cell functions? Differentiation. It has to undergo to differentiation and finally form a tissue. So the cell count and viability at different time after administration is also very important, so that we can identify, okay, for example, now imagine I have seeded the cell in the scaffold or a biomaterial. So now at 0 hour, what are the number of cells? Okay, what is the cell viability? That means how exactly the cells are functioning or not, or whether they are active or not, whether they are functioning or not, whether they are living or not. So that is at 0 hours. Next after 10 hours, next after 20 hours. Like that I can have a interval, different interval at different time after the administration. So that is also very important role, so that we can check whether the biomaterial is supporting for cell division or not, whether the cell numbers are increasing or not, and whether the cell is living for that so much amount of duration or over a period of time is or not. So that itself is the cell number and cell viability understanding. That's how we can have an example. So next we are having cell count or cell number, that is usually we with respect to the instruments, and also we can count the cells. So for usually we use hemocytometer. So hemocytometer is also it's called as a Nebular's chamber. So it aids to the counting of the cells. That means you will be using hemocytometer. I guess most of the biotechnology department will be having the hemocytometer, where we will be used to count usually the colonies or the cells or something like that. So a sample of homogeneous suspension of cells should be loaded. A particular sample you take, that should be obviously homogeneous suspension, that should be loaded into the chamber, and the cells are counted under the microscope. So manually you can count how this particular hemocytometer will have a grid that will have a different scales. So now if I am viewing with this particular the cube or that scale, so much number of, imagine you have to check only for if the cells are distributed uniformly. Since the homogeneous solution is there, that has to be cells should be distributed uniformly. So when I check in one box, if in one box there are 25 cells are falling, okay, I have to check only one box. Now multiply with the number of boxes or which the cells can be covered, right? So that's what is explained here. A sample of homogeneous suspension of cells should be loaded into the chamber, and then the cells are counted under the microscope manually and multiplied with a scaling factor. So if there are 25 scale, if you see if it is a square kind of flick, imagine cells are there like around 75%. So if I am having around 100 squares, so I will be taking about 75 squares only wherever the cells are there. So just count the number of the squares and count the number of the cells in one square. So that will into the number of cells. So here it is 25 into 75. So that will give you the total number of the cells, right? Multiplied with the scaling factor. That scaling factor is here is 75 in my example to yield the cell number in the suspension. Now this technique can be used in combination with a trypan blue stain. So you can also, along with this, you can also use this blue stain, trypan blue stain. When you use this particular stain, what happens, which was based on the exclusion principle, that means it differentiates living cells from the dead cells, right? So whatever the living cells will be there, that will take up the one particular color, and the others dead cells will take up another color. So we can differentiate, we can count only if it is only blue cells are there, that means those cells are living cells. So I can count only those living cells, so I can avoid the dead cell count. Okay, in normal, without staining, even dead cells also you will be counting. With the staining, you will count only living cells. So it will not there will not be much error. Okay, so next we are having Calder counter. So that can also be used for counting cells, although this electronic counter results may be erroneous if the cells are aggregated. That means in this particular next approach we are having as a counter, that is Calder counter. In Calder counter, what happens? You can use it for the cell counting, but what happens if any cells are aggregated, then it will not count. Imagine 3 cells are aggregated, it will count it as only 1 cell. Whereas in manually we can differentiate it, whereas in staining we can differentiate it. Next, cell viability. That was about the cell number. Next, cell viability. So there are other indirect methods are there for assessing the cell viability and as well as count. Now we know what is cell viability. That means ability of a cell, okay, to live or to do function over a period of time. So these are based on the DNA content, so protein content, and mitochondrial or metabolic activity using fluorescence, calorimetric or enzymatic conversion principles. Obviously, the method, whatever the method you are using it to check the viability and count, that depends on what you are talking about. I mean, what the material or what kind of approach you are using. The most commonly employed tests followed is you are having MTT and MTS assays, okay, in laboratories. So MTT and MTS is nothing but they are chemical names. You can just use them as like indicator kind of names, right? MTT stands for we are having 3,4,5 dimethyl thiazole, whereas we are having the same thing goes like this again, 2,5-diphenyl tetrazolium bromide. So this is nothing but one MTT, and this MTT stain, so is it's actually is a yellow tetrazole which gets reduced to the purple pharmacin by live cells. That means when you add this particular MTT, okay, into the solution, that homogeneous solution of cells, so only the yellowish yellow color of the tetrazole will be converted into the purple color when they bind with the living cells. So only living cells will have that purple color, other dead cells will be having different color. Or so you can see that these particular cells are living. You can separate the living cells with the dead cells. So MTS is nothing but again 3,4,5 dimethyl thiazole, 2-YL, 5,3 carboxymethoxy phenyl, 2,4 sulfoxyphenyl, 2H tetrazolium. It's in the presence of the phenazyme methosulfate, that is again one more compound you have to add, that is PMS, that this particular this will produce a pharmacin product. That means if the living cells are there, it will produce the pharmacin products. So these conversions either in MTT or MTS assay, that takes place only in living cells, or else it will not happen. That means here pharmacin product will be formed if the living cells are there, if dead cells cannot produce it. So hence the change in the absorbance when using MTS and MTT is used as the indirect method for the measuring cell viability. So whenever you are seeing the difference in color, so for what technique we are using, characterization technique we are using EV spectrophotometer, where we can check the absorbance at different wavelengths, and we will come to know that by measuring the wavelength or by measuring the absorbance, we can differentiate. It can be used as indirect approach to check whether the cell is living or not, whether its living cells are there or not. That if the cells is living means they are doing, or definitely they will do their function. So that's it. That was about the cell number and viability, cell count and viability. Next topic we are having cell motility and migration, that we will discuss in the next class.